Hello everyone, my name is Dr. Ambud Misra, and in this video we are going to discuss about the process of beta oxidation, ketogenesis, ketosis, diabetic ketoacidosis, and some diseases related to it. So in the previous video we discussed about the process of lipolysis, where we saw that how a triacyl glycerol is going to broken down into glycerol and a free fatty acid. So we have already seen that this here is your enzyme adipocyte triglyceride lipase. This is hormone sensitive lipase, and here you have monoacyl glycerol lipase. So in turn we are going to get fatty acid 1, 2 and 3 and one molecule of glycerol. Now what is the fate of glycerol? So this glycerol is going to get broken down further into glycerol 3 phosphate, and which further is going to broken down into dihydroxyacetone phosphate. And further there are two fate of this dihydroxyacetone phosphate. The first one is this, either this is going to enter into the process of glycolysis, or it can further go back into the process of synthesis of triacylglycerol and phospholipids. So here synthesis of triacylglycerol. So the fate of dihydroxyacetone phosphate is it is either it is going to form, it is going to enter into the process of glycolysis, which further is going to enter into TCA cycle and generate energy, or it can go back into the formation of synthesis of triacylglycerols. Now you see what is the fate of free fatty acid. Now this free fatty acid that formed inside the adipocytes as a result of lipolysis is further is going to enter into the circulation bound to albumin, and when it is being taken up by the cells, it is going to bound to fatty acid binding proteins, and which in turn is going to enter into the process of beta oxidation. And this beta oxidation in turn is going to provide energy. Now further diving into the topic of beta oxidation proper. So there are three steps in the process of beta oxidation. First one is the activation of fatty acid, then the transport of fatty acid, and beta oxidation proper. So activation of fatty acid means the fatty acid is going to get converted into acyl coenzyme A, that is your activation of fatty acid. And this process is going to occur inside the cytosols. This process is going to occur inside the cytoplasm. And in order to proceed the process of beta oxidation, this acyl coenzyme A needs to get inside the mitochondria. So transport of fatty acid means it is going to happen from the cytosol into mitochondria. Then comes the main step, that is your beta oxidation proper. This is obviously is going to happen inside your mitochondrial matrix. So there are three steps in the process of beta oxidation. Now we will discuss each of this one by one in details. So what happens in the activation process? In the activation, this free fatty acid is going to get converted into acyl coenzyme, is going to get converted into acyl coenzyme, and the enzyme that is responsible for this is your thiokinase, and this is also known as your acyl coenzyme synthase. These two are the same. This is the two names of the same enzyme. Plus what other intermediates are required for this process is your ATP, your coenzyme, and one ion, that is your magnesium ion. So this is the summary of activation of free fatty acid, which when it is going to get converted into acyl coenzyme with the help of thiokinase, or acyl coenzyme A synthase with the help of some other intermediate, that is your ATP, coenzyme A, and magnesium ion. Now this acyl coenzyme is present inside the cytosol, and for for the process of beta oxidation to proceed, this acyl coenzyme needs to get inside the mitochondrial matrix. So the second step of your beta oxidation is your transport of this acyl coenzyme A from cytosol into the mitochondrial matrix. So how this is going to happen? Cytosol has a reserve of carnitine, and this carnitine is going to help incorporate this acyl coenzyme from cytosol into the mitochondrial matrix. So this is going to happen. This is your acyl coenzyme, and this is going to get combined with carnitine. This carnitine is already present inside the cytosol. Cytosol has the reserve of carnitine. Now when acyl coenzyme combines with carnitine, this is going to form acyl carnitine plus coenzyme A. Now this acyl carnitine is now going to get transferred inside the mitochondrial matrix, and this carnitine is from cytosol reserve, and the enzyme responsible for this reaction to carry out is your CPT1, that is your carnitine palmitoyl transferase 1. This is an important enzyme, your CPT1. Now this acyl carnitine, when it enters mitochondrial matrix, your acyl carnitine, now further this is going to combine with coenzyme. The reserve of coenzyme is already in mitochondrial matrix, and further this is going to form carnitine plus acyl coenzyme. So this acyl coenzyme is now has been incorporated inside the mitochondrial matrix. The enzyme responsible for this is your CPT2, that is your carnitine palmitoyl transferase type 2, and this carnitine is further going to get back into the cytosol so that it can be reused. So this is the process of transport of acyl coenzyme from cytosol into the mitochondrial matrix. The enzyme that is present in cytosol is your CPT1, in mitochondrial matrix is your CPT2, and the reserve for carnitine is present in cytosol. The reserve for carnitine is also there in mitochondrial matrix, but the carnitine which is used is provided by cytosol. So this is the process by which acyl coenzyme is back. This is how acyl coenzyme goes into mitochondrial matrix, and carnitine goes back into the cytosol. Now this is known as carnitine subtle. It's also called carnitine subtle. This is called it carnitine subtle. Now there is one inhibitor of CPT1, or you can say carnitine subtle as a whole, that is your malonyl coenzyme. This malonyl coenzyme is the inhibitor of CPT1 or carnitine acetyl, and this malonyl coenzyme, this is the metabolite, one of the metabolite of fatty acid synthesis. The malonyl coenzyme is one of the metabolite of fatty acid synthesis. So that means if the process of fatty acid synthesis is going on, the process of beta oxidation cannot occur, because malonyl coenzyme, which is formed during the process of fatty acid synthesis, is going to inhibit the process of beta oxidation, or you can say it is going to inhibit carnitine acetyl, which is a really important step for the process of beta oxidation to occur. So at one point of time, beta oxidation and fatty acid synthesis cannot occur, and this is the reason, that is your malonyl coenzyme. So this is the metabolite of fatty acid synthesis, and means when fatty acid synthesis occurs, beta oxidation cannot occur. So this is the reason that fatty acid synthesis and beta oxidation cannot happen simultaneously. So we saw activation where fatty acid got converted into acyl coenzyme. In the second step, that is the transport, acyl coenzyme transported from cytosol into the mitochondrial matrix. The enzymes involved are CPT1 and CPT2, and carnitine plays a very important role. And the inhibitor of carnitine acetyl or transport of fatty acid, or you can say inhibitor of CPT1 is your malonyl coenzyme, which is the metabolite of fatty acid synthesis. Now comes the third step, that is your beta oxidation proper. So where this acyl coenzyme that we got from the first step and which came inside the mitochondrial matrix is going to get combined with one molecule of coenzyme, and which further is going to form one acyl coenzyme, which is 2 carbon lesser in number, plus acetyl coenzyme. This acetyl coenzyme is further going to enter Krebs cycle, and then it is going to generate the energy. So this acetyl coenzyme is going to enter Krebs cycle, which further will generate energy, and which can be further used by the different organs and the body. Now what is the intermediates and enzymes that are required for this beta oxidation proper is your FAD and NAD. This is going to get converted into FADH2, NADH, and H plus. So this is also required for this process, and there are 4 enzymes that are involved in this step, but we just need to remember 2 enzymes. The first one is which is required in the first step of beta oxidation proper, is your acyl coenzyme dehydrogenase, which is required in the first step, and another one is your thiolase, which is required in the last step of beta oxidation proper. Now you must have got confused about this coenzyme, acyl coenzyme, acetyl coenzyme. So what are these? So coenzyme, it is the carrier of acyl group, and this coenzyme, this is the carrier of acyl group. And what is the functional group that is present in the coenzyme? That is your SH, this group, which is your sulfur hydryl group. This is known as sulfur hydryl group. So coenzyme is nothing but the carrier of acyl group, and it has the functional group, that is your sulfhydryl group. So via which the acyl group is going to get attached to coenzyme. Now, what is the difference between acyl coenzyme and acetyl coenzyme? See, you must be familiar with the structure of a fatty acid that I told you in the previous class. So you see, fatty acid is RC double mondo H. That is your carboxylic acid. This are, or you can say this, or you can say this portion. Remove the H, or so this thing is going to get attached to coenzyme, forming acyl coenzyme. So acyl is a general term, okay, and acetyl is your this group. See, acetyl is this group, and acyl is this group. So this R comes from the fatty acid. This R comes from fatty acid. This is your acyl coenzyme. This is your acyl group. This is your acetyl group. This R is going to come from the R that is present in fatty acid, and acetyl means this R is your CS3. That's it. That is the difference between acyl coenzyme and acetyl coenzyme. So you can say acetyl coenzyme is a subtype of acyl coenzyme. So just to clear the confusion between acyl and acetyl. Now the two important enzymes are acyl coenzyme dehydrogenase and your thiolase. Now another one disease that need to be mentioned here, that is your Jamaican vomiting sickness. So what happened in this disease, you will see, it is going to happen when a person consumes a fruit that is named ackee fruit. This is your ackee fruit. This fruit is going to block enzyme acyl coenzyme dehydrogenase. This enzyme, which is the first step of beta oxidation proper, this enzyme is going to get blocked when the person consumes ackee fruit, and when this enzyme is going to get blocked, this will in turn going to block the process of beta oxidation proper, or in as a whole the beta oxidation. So blocked beta oxidation. So beta oxidation is blocked. So what are the clinical features that you are going to see in this? You will see the clinical features: hypoglycemia, convulsions, coma, and even death. So in Jamaican vomiting sickness, enzyme that is going to blocked is your acyl coenzyme dehydrogenase, which is required in the first step of beta oxidation proper. It happens due to consumption of ackee fruit, and which is going to block beta oxidation. So there will be accumulation of fatty acids, because fatty acids are not going to oxidize through beta oxidation. So there will be accumulation of fatty acid, and the clinical features are your hypoglycemia, convulsion, coma, and death. Now, what is the fate of this acetyl coenzyme? This acetyl coenzyme, you see, it's the one of the fate is it is going to enter Krebs cycle, and which in turn is going to generate energy. But this acetyl coenzyme can go through other pathways, to give some other products, or can enter into another in synthesis of other products. Now what is the fate? Like there are multiple. The one of the main the most important is your entering enter scrap cycle and leads to energy. Another one is this is going to get back into the process of synthesis of fatty acid and then triacyl glycerol. So it can also enter into process of lipogenesis, which will form fatty acid and then triacyl glycerol, and which in turn again is going to accumulate inside the adipocyte or stored inside the adipocytes. Another function, another fate of acetyl coenzyme is your ketone body synthesis. Ketone body synthesis. And the next one is your cholesterol synthesis. Cholesterol synthesis. So acetyl coenzyme can enter Krebs cycle by thereby generating energy. It can also enter into the process of lipogenesis, forming fatty acid and then triacyl glycerol. It can also enter into the process of ketogenesis, forming the ketone body. That is your ketogenesis. And it can also enter into the process of forming cholesterol. That is your cholesterol synthesis. And further, this cholesterol is going to enter into steroidogenesis, thereby forming steroids, your steroid hormones. So this acetyl coenzyme, that is the end result of beta oxidation proper, has 4 fates. That is your entering Krebs cycle, generating energy, lipogenesis, ketogenesis, and cholesterol synthesis. Now let's discuss about the process of ketogenesis. So this process is going to happen inside the mitochondrial matrix. Same as beta oxidation proper. Now ketogenesis, the major contribution for the ketogenesis comes from this acetyl coenzyme that is formed in the process of beta oxidation, but it can also form from other processes also. So that is the major constituent is your acetyl coenzyme from beta oxidation, but some other are that we are going to see here is your from pyruvate. Ketogenesis can also occur from pyruvate, and it can also occur from some amino acids. That is your leucine and lysine. So leucine and lysine, these are exclusively ketogenic. Like they are going to generate ketone bodies. There are some other amino acids that are not exclusively ketogenic, but these two are exclusively ketogenic. These are exclusively ketogenic. That is your leucine and lysine. But the important one is from acetyl coenzyme. Now, how this process is going to occur, that is how this acetyl coenzyme is going to form ketone body that we are going to see here. So when two acetyl coenzyme, like two molecules of acetyl coenzyme combines, this is going to form acetoacetyl coenzyme, and the enzyme responsible for this is your beta ketothiolase. Now again further, this acetoacetyl coenzyme is going to combine with one molecule of coenzyme, and the enzyme responsible is your HMG coenzyme synthase. HMG coenzyme synthase, which is going to form HMG coenzyme. What is HMG? HMG stands for, this is 3-hydroxy 3-methyl glutaril. Nothing important, just for the sake of knowledge. So acetoacetyl coenzyme further combines with one molecule of coenzyme with the help of HMG coenzyme synthase is going to form HMG coenzyme. Further, this HMG coenzyme is going to is going to get lysis and going to form acetoacetate. So this is your HMG coenzyme lyase. HMG coenzyme lyase, and here one molecule of acetyl coenzyme is going to get released. Now this in turn is going to form acetoacetate. Acetoacetate is your, this is your you can say central precursor for ketone bodies, or you can say this is your primary ketone body. This is your primary ketone body. This acetoacetate can be converted into acetone or beta hydroxy butyrate. See, this acetone, this process is transient. Acetoacetate can convert into acetone, and it can further convert into acetoacetate. This is the transient process, and this acetone is volatile. This is volatile and it has no use. It is going to get out from the body via lungs or from the urine. So this is going to get excreted via breath or urine, and does not form or generate energy. So acetone is a volatile compound which is going to excreted via breath or urine, and it does not generate any kind of energy. Plus this acetone is going to give us some particular smell from the breath, that is your sweet fruity smell. So whenever you see like sweet fruity smell from breath, we are talking about acetone. Now this beta hydroxybutyrate, this is your this is going to involve in the process of generation of energy. So this is important. Acetone has no significance at all in the process of generating energy, or it has no significance in generating any other compound. But acetoacetate and your beta hydroxybutyrate, this is going to generate energy. Now further, some things about ketone body. So ketone body, it is water soluble, and it is formed in liver. It is formed not exclusively, but mostly it is formed in liver. The ketogenesis occurs inside the liver. Now this ketone body, or which is formed, is transported to various organs from liver, and it is utilized by the organs which have mitochondria. So the process of ketogenesis occurs inside the liver, and further this ketone body that has been synthesized inside the liver is going to transported to various organs from the liver, and it is going to get utilized by the organs which contains mitochondria. Now, it is used as energy source when glucose is in short supply. So you see, when some conditions, like certain conditions where the body is deficit in glucose, so the main source of energy is your ketone body. So what are the cases, what are the situations, or what are the, you know, conditions you can remember where the glucose is in short supply? One you can say is starvation, when the body is not consuming any kind of food, and in that case also carbohydrates. And in case of diabetes mellitus, in diabetes mellitus, glucose is present inside the body, inside the blood, but this glucose cannot get incorporated inside the tissues, inside the cells, because machinery is, you know, not working properly. Either insulin is not forming, or the cells are not picking up a glucose that is present in the bloodstream. So the two conditions where uh, ketone body is going to form in excess is your starvation and your diabetes mellitus. In these two conditions, uh, the glucose is in short supply, and the main source of energy is your ketone body. One more thing, this ketone body synthesis is a continuous process. It is occurring inside the healthy body also. It's not like it is going to form only when the glucose is deprived, but this ketone body synthesis is very minimal. Like you cannot see, you know, a huge amount of ketone body present inside the bloodstream, or is excreted through the urine. So this process is continuous, but ketone body that is formed in normal healthy individual is very less, and it becomes prominent whenever the in certain conditions where the glucose is in short supply. Now, what is ketosis? You have heard about this term, ketosis. So ketosis is there are two conditions combined which can in turn lead to ketosis. That is your ketonuria and ketonemia. When there is more ketone inside the urine and more ketone inside the blood, these two together combined is known as ketosis. So ketonuria means increase ketone in urine, and ketonemia means increased ketone in blood. So these two combined is known as ketosis. That is, your body has more ketone bodies present inside the bloodstream and your urine. Now, the smell of acetone in breath is we saw it is it's like a sweet smell or fruity smell. See, and it is seen in again your starvation and your diabetes mellitus. Starvation in the sense in the late stage of fasting, like let's just say if the person is fasting after 48 hours, then also like it comes in under late fasting. So after 48 hours of fasting, the ketosis is going to appear inside the body. Now you see, we have seen that when the glucose is in deficit, there is less glucose inside the body. The body diverts into the forming of ketone bodies, which in turn is going to generate the energy which glucose cannot. So how this ketone body going to generate the energy, we will see about it. So let's just say acetoacetate and beta hydroxybutyrate, these are also they are like interchangeable. Like acetoacetate is going to form beta hydroxybutyrate, and vice versa. So this acetoacetate is going to form acetyl coenzyme, two molecules of acetyl coenzyme, which further is going to enter Krebs cycle, and which will in turn generate the energy. So again, the whole, the the main goal of ketone body is again to form acetyl coenzyme, which is in turn going to enter Krebs cycle and generate energy. So and the enzyme that is responsible for this is your thiophorase and thiolase. These two enzymes are responsible for this process. This is the summary of how ketone body is going to generate energy. The ketone body again is going to form as two molecules of acetyl coenzyme, which in turn is going to enter into the process of Krebs cycle, which is the ultimate source of generating energy inside the body. Now, you see, there is one diagrammatic representation that you must have a rough idea about. So this fatty acid, when the beta oxidation occurs, the fatty acid is going to convert into acetyl coenzyme, and glucose, when enters glycolysis, it is going to form pyruvate, and further by pyruvate dehydrogenase, this is going to form acetyl coenzyme, and this acetyl coenzyme is going to enter TCA cycle and generate energy. So the ultimate source, ultimate process through which the body is going to generate energy is your Krebs cycle, TCA cycle, and whichever compound is going to generate energy has to enter Krebs cycle one way or another. So there is one intermediate compound molecule, that is your acetyl coenzyme. This is going to form in every in every case, like in case of beta oxidation, in case of glycolysis, and again in case of ketogenesis. All these processes, these are going to form acetyl coenzyme, which in turn is going to enter Krebs cycle and to generate energy. So this fatty acid enters beta oxidation, forming acetyl coenzyme. Glucose enters glycolysis, forming pyruvate, then through via pyruvate dehydrogenase, the three enzyme complex, this is going to again form acetyl coenzyme. And again this process, acetoacetyl coenzyme, acetoacetate, this is your ketone body, and again this is going to form acetoacetyl coenzyme, and which in turn is going to form acetyl coenzyme. So acetyl coenzyme is your primary molecule, you can say, that is going to or you can say the intermediate or common molecule that is common in all these processes. So all these processes has to generate this molecule in order to generate energy, because this is the molecule that is going to enter the Krebs cycle. So in glycolysis, in beta oxidation, in ketogenesis, in every process, acetyl coenzyme is going to get generated, which is going to in turn going to enter into the Krebs cycle to generate energy. So this is a line diagram that you can remember. This is really helpful. Now coming into the process of starvation, what happens actually in the starvation? So to maintain the blood glucose, see, whenever the body is deficit of glucose, so there must be a way to maintain this blood glucose level. But even though you maintain this blood glucose level, or the body tries to maintain the normal level of blood glucose, the body is not able to generate energy from that glucose. So to maintain blood glucose in initial phase, that is your after for 24 to 48 hours of taking food, the main source of blood glucose is your glycogenolysis. You see, the body has the store of glycogen. So in this phase, in 24 to 48 hours of fasting, the body generates glucose from the process of breaking down the glycogen. That's how body gets the glucose. But after 48 hours, this process is going to happen through the process of gluconeogenesis, because now the stores of glycogen are depleted. So there is no way that a body can maintain normal blood glucose through the process of glycogenolysis. Now the body needs to find any other way in order to maintain the blood glucose. So body does this by the process of gluconeogenesis. And you see, if you have studied or learned about the process of gluconeogenesis, the intermediate, that is your oxaloacetate, this intermediate is necessary for the process of gluconeogenesis. And this oxaloacetate is also used. So for now, let's just say, like, the store of oxaloacetate is used up through by the process of gluconeogenesis. See, and that is your, after this comes your starvation. Now, when the glucose is deficit, body needs to do two things. First, it needs to maintain the blood glucose. Second, it needs to generate energy. So the first thing, the primary focus is to maintain the blood glucose. That is done by the process of gluconeogenesis in the phase of starvation, and it utilizes oxaloacetate. Now, in order to generate energy, now this glucose is cannot be used up to generate energy, right? Because body has done lots of effort to generate this glucose to maintain the blood glucose level, and again body cannot use this glucose to generate energy, because what is the point of generating glucose then in the phase of starvation, if ultimately this glucose is going to get used up in the production of energy. So as the body has other ways to generate energy, that is your ketogenesis through the ketone bodies. So this gluconeogenesis is responsible for maintaining blood glucose, and to get energy, body uses fatty acid degradation from the adipose tissue, and which in turn causes overproduction of ketone bodies, or you can say overproduction of acetyl coenzyme. Now see, this acetyl coenzyme, how fat generates energy. Fat generates energy by the process of lipolysis, and then by the process of beta fatty acid degradation, that is your beta oxidation, acetyl coenzyme is going to get formed, and this acetyl coenzyme is going to enter TCA cycle, Krebs cycle, which in turn is going to generate energy. But in order to run Krebs cycle, oxaloacetate is an important intermediate that must be available to acetyl coenzyme to generate energy through Krebs cycle. But this oxaloacetate is going to get used up in the process of gluconeogenesis. So this oxaloacetate is not available to this acetyl coenzyme, so that this acetyl coenzyme can enter Krebs cycle and in turn generate energy. So the stores of oxaloacetate is depleted already, because this is used up in the process of gluconeogenesis. So this acetyl coenzyme cannot generate energy, because it ultimately cannot enter the Krebs cycle. So what happens? There is accumulation of this acetyl coenzyme, and this is in turn diverted to the production of ketone bodies, which in turn is going to cause ketosis. So that's how ketosis is going to occur in case of starvation. I hope what I was trying to tell you, I hope you guys understood. See, the whole point is, there are two things. When the body is starving, body needs to maintain blood glucose, and body needs energy. Body maintains blood glucose, and it does so by the process of gluconeogenesis, where it is uses oxaloacetate. So in order to get energy, another option is to break down fatty acid by beta oxidation, and beta oxidation generates acetyl coenzyme. But this acetyl coenzyme needs to get back into Krebs cycle, but this needs this intermediate, that is your oxaloacetate, which is not available because it is used up by the process of gluconeogenesis. So this oxaloacetate is in deficit, and this acetyl coenzyme A that is generated through the beta oxidation has no other option but to form ketone body. So that's why in starvation there is excess of ketone bodies, which in turn is going to cause ketosis. Now you see, ketone body, it is under constant production and utilization in the normal condition. The normal concentration of ketone body in blood is your like mostly like it is around 1 milligram per deciliter, or you can say like even lesser. And in normal conditions, the excretion in urine, the excretion of ketone bodies in urine is negligible, and this is detected by Rothera's test. Rothera. The ketone body in urine is detected by Rothera's test. So in normal condition, it is under constant production and utilization. The normal concentration is even lesser than 1 milligram per deciliter, and the excretion of ketone bodies in urine is negligible in the normal condition, and it is detected in the urine. The ketone bodies in the urine is detected by Rothera's test. This is your R, Rothera test. Now starvation, we see ketosis, and we will see ketonemia and ketonuria, where there is increased concentration of ketone bodies inside the blood and inside the urine. Now you see, we are going to discuss about keto diet, ketogenic diet, or you can and ketonuria, related ketonuria and weight loss. So what is keto diet? Like the keto or ketogenic diet is almost like the similar, like the same meaning. It has the same meaning. So you must have heard about this keto diet. So what is the significance of keto diet? So in case of in keto diet, you are going to see the amount of fat is maximum, protein is moderate, and carbohydrate is minimum. See, it is a high fat, moderate protein, and low carbohydrate diet. So why it is significant to take more fat if you have to lose weight? See, now comes the significance of taking high fat diet. So what happens in high fat diet? If you take high fat diet, there is more fat available. The more fat goes into the process of beta oxidation, and when the more fat goes into process of beta oxidation, again similarly, there you are consuming less carbohydrate. The carbohydrate consumption is less, so blood glucose will be maintained by the process of gluconeogenesis or glycogenolysis, or you can say this less carbohydrate that is available. So most of the glucose that body is generating is through other processes, and which is again going to use oxaloacetate, and oxaloacetate is going to get depleted. So what is going to happen? The body is trying to break down fat, because for for generating energy, the carbohydrate option is not available, because there is very minimal carbohydrate that you are consuming, and also the body need to maintain the blood glucose. So body shifts for the need of energy from carbohydrate to fat, and when it shifts its requirement for energy to fat from carbohydrate, it is going to generate more acetyl coenzyme, and this acetyl coenzyme is going to form more ketone body, and this ketone body and this acetyl coenzyme, this whole is going to generate energy. So I hope you understand what I'm trying to say. It is similar to starvation, and when this body is the body is going to break down fat that you are consuming. So you can also say that if you are consuming more fat, so body is going to break down that fat. But what happens in, in long term, the body shifts its energy demand from carbohydrate to fat, and is this fat also include the fat that is stored in your adipocytes. So it is also going to break down that fat that is in your adipocyte along with the fat that you are consuming. So this more fat, the significance of consuming more fat is only to shift the energy demand from carbohydrate to fat, and that fat also includes the fat that is stored inside the adipocytes. I hope you get it. So, less glucose is available for energy, and it shifts, energy demand from glucose to fat, as energy source. Now, dietary fat is broken down along with stored fat in adipose tissue. So, this is going to produce ketone body, and this ketone body is used as your energy source. So, now in keto diet, in ketogenic diet, you are going to see ketonuria. Why you are going to see ketonuria? Because there is, more ketone bodies that are going to get formed, and if more ketone bodies are there, if ketosis is there, there will be more ketone bodies present inside the urine. So if you see that if someone is doing keto diet and there is ketone body inside present, significant amount of ketone bodies in the urine, that indicates, that indicates active fat metabolism. Ketonuria in keto diet indicates active fat metabolism. Now, breakdown of stored fat, in adipose tissues, it is going to contribute to your weight loss. This is going to contribute to your weight loss. Now, you see, when the less carbohydrate, if you are consuming, less carbohydrate, that means your insulin is going to get released when there is more carbohydrate, right? So when you are consuming less carbohydrate, so less insulin is going to get released, and this less insulin is going to contribute to fat breakdown. In last video, you saw that the process of lipolysis, the process of lipolysis is increases, the lipolysis increases when there is more insulin, less insulin, sorry. So like insulin inhibits the process of lipolysis. So if there is more insulin, the process of lipolysis is going to get inhibited. So when there is less insulin, this is going to promote the process of lipolysis. This is in turn is going to promote fat metabolism, and which in turn is going to contribute to your fat loss. So this whole thing about keto diet or ketogenic diet is and how this in turn is going to contribute to weight loss is this mechanism, like shifting from carbohydrate to fat breakdown, and that also contribute the fat that is stored inside the adipocytes. Now, so it is helpful in, again, weight loss. Now, what happens in diabetes mellitus? In diabetes mellitus, you see there is carbohydrate metabolism which is going to get decreased. So as there is less carbohydrate available to the tissues, and the less energy that is obtained from these carbohydrates. So to meet the energy demands, like as I earlier described, there will be increased lipolysis. Increased ketone body production, and which in turn is going to contribute to ketosis in case of diabetes mellitus. You see, in diabetes mellitus, there is decreased metabolism of carbohydrate. When there is decreased metabolism of carbohydrate, there will be less carbohydrate available to the tissues. And when there is less carbohydrate available to the tissues, the energy generated from the carbohydrate is going to be very less. So to meet the energy demands, the body needs to uh, do lipolysis in order to meet the energy demands. So if the body is going to do lipolysis, there will be more ketone bodies that will be produced. Again, you will say, like, why this acetyl coenzyme that is going to form in the process of lipolysis is going to generate energy without forming ketone body. So then comes in the picture your oxaloacetate, which is your intermediate and very important for, which is really important for Krebs cycle to occur. And if this oxaloacetate is in deficit, with whom this acetyl coenzyme is going to get combined in order to enter TC cycle. So that's the reason this acetyl coenzyme from beta oxidation is not available for energy, and which in turn is going to produce ketone body, which in turn is going to cause ketosis. Now, what is diabetic ketoacidosis? This acetoacetate and beta hydroxybutyrate. See, these are going to contribute to acidosis. How? See, these both are strong acids. Why? Because they both contain carboxyl group. They contain carboxyl group. So they both act as strong acids. And so what is the property of a strong acid? A strong acid means whenever it goes into a fluid, into water or into blood, it is going to get dissociated. So it is going to get dissociated, release H plus, and pH is going to get decreased, which is going to cause acidosis. That is the significance of diabetic ketoacidosis. So these ketone bodies, that is your acetoacetate and beta hydroxybutyrate, these are strong acids. They contain carboxyl group, and they are going to get dissociated the moment they reach inside the blood. And when they dissociate, they release H plus, which is going to decrease the pH, and which further is going to cause acidosis. So in case of uncontrolled diabetes, you are not going to see in case of controlled diabetes. You are only going to see this in the diabetic ketoacidosis in the case of uncontrolled diabetes, and this can result in coma or even death. And the treatment for diabetic ketoacidosis is your insulin, just to manage the blood glucose. So this is about diabetic ketoacidosis. Now we will discuss some disease related to it. One is your Refsum disease. What is Refsum disease? So in Refsum disease, there is defect in the process of alpha oxidation. And how the alpha oxidation is get defected? Because this is going to it's going to suppress this enzyme known as phytanoyl coenzyme hydroxylase. So in Refsum disease, there is defect in alpha oxidation that is due to defect in the enzyme phytanoyl coenzyme hydroxylase. What is alpha oxidation? Like we have seen the beta oxidation. So alpha oxidation occurs in order to break down branched chain fatty acids. Because why these fatty acids cannot be broken down by the process of beta oxidation? Because you see in branched chain fatty acids, the beta carbon contains methyl group. That means it is not free. So beta oxidation mainly occurs on the beta carbon. So as the beta carbon is occupied by the methyl group, so beta oxidation cannot occur in case of branched chain fatty acids. So there occurs alpha oxidation. So alpha oxidation, it is going to generate no energy. It generates no energy. Alpha oxidation generates no energy, and this process is going to occur inside the peroxisome. So this enzyme is present inside the peroxisome. So there is accumulation of branched chain fatty acids inside the peroxisomes. Now, what are the clinical features that you need to remember? That is your retinitis pigmentosa, your ichthyosis, where you are going to see scaly fish like skin. Now another one is your peripheral neuropathy and arthralgia. Or you can say phytanic acid. There is accumulation of branched chain fatty acid. The most important branched chain fatty acid that is going to get accumulated is your phytanic acid, because the enzyme defected is your phytanoyl coenzyme hydroxylase, which is the enzyme responsible for the metabolism of phytanic acid. So this phytanic acid, or which is a type of branched chain fatty acid, is going to get accumulated in case of Refsum disease, where the alpha oxidation is defected. Clinical features are retinitis pigmentosa, ichthyosis, peripheral neuropathy, and arthralgia. And the treatment of this is restrict dietary product and green leafy vegetables, because these contain this phytanic acid. Now next disease is your Zellweger syndrome, or you can say cerebrohepatorenal syndrome. Here what happens, here the defect is your peroxisomal targeting disorder. See, the enzymes which are synthesized inside the peroxisomes, the enzymes that are synthesized inside the endoplasmic reticulum, they need to go inside the peroxisomes. So this process is done with the help of peroxisome targeting sequence. So there is this PTS, that is your peroxisome targeting sequence, with help of which the enzymes that are synthesized in smooth endoplasmic reticulum goes into peroxisomes, and that is done by PTS. So when peroxisome targeting sequence is defective, this disorder is known as peroxisome targeting disorder, because peroxisome targeting sequence is defected, and the enzyme that is present inside the smooth endoplasmic reticulum is not able to go inside the peroxisomes. So this in turn is going to affect oxidation of, or you can say there is defect in oxidation of very long chain fatty acid and branched chain fatty acid. So in case of Zellweger syndrome, peroxisomal targeting sequence is defected, that causes peroxisomal targeting disorder, and the enzyme that is formed inside the smooth endoplasmic reticulum is not able to go inside a peroxisome where it is required. So there will be defective oxidation of very long chain fatty acid and branched chain fatty acid, because the oxidation of these fatty acid occurs inside the peroxisome, because the enzymes that are required for the oxidation of these is present in peroxisomes, but due to this defect these cannot come into peroxisome from a smooth endoplasmic reticulum. So there will be accumulation of there will be accumulation of very long chain fatty acid and branch chain fatty acid. What are the clinical features that you are going to see in this case? You are going to see mongoloid facies, up slanting, palpebral fissure, up slanting palpebral fissure, frontal bossing. You are going to see frontal bossing or high forehead. The most important is your brush field spots in your iris. You will also see epicanthal folds, and this is going to resembles Down syndrome. This feature is going to resemble Down syndrome. The brush field iris spot is your spotter. So whenever you are going to see brush field iris spot, it is your Zellweger syndrome or cerebrohepatorenal syndrome. The defect is in your peroxisomal targeting disorder sequence, which causes peroxisomal targeting disorder. The defect is in the oxidation of very long chain fatty acid and branch chain fatty acid, which causes accumulation of these following. And the clinical feature are mongoloid facies, upslanting palpebral fissure, frontal bossing, high forehead, brush field spots in the iris, and epicanthal folds. And these facial features and these clinical features is going to, appear like is going to resemble same as Down syndrome. So this was about today's discussion. In the next video, we are going to discuss about glycolipid, phospholipids, and some lysosomal storage disorders, and some sphingolipidosis, and all these. So thank you for watching today's discussion. Have a nice day.